Welcome to Micro Review. Today our topic of discussion would be Bacillus, and Bacillus is having two species. One is the Bacillus Anthracis and one is the Bacillus cereus. First topic of discussion would be Bacillus Anthracis and the second topic would be Bacillus cereus, right? First of all, we must be knowing that under which headings we would be particularly discussing a bacteria. First would be the disease it causes, second would be the features, then the transmission, then the pathogenesis, then the clinical findings, then the lab diagnosis, and then the treatment and the prevention. So let's go on with the Bacillus anthracis. It causes anthrax. The anthrax is actually of three types. One is the pulmonary anthrax, the cutaneous anthrax, and the GIT anthrax. If we talk about the features of Bacillus anthracis, it is actually a gram-positive rod, which everyone knows. The main point of differentiation here is that it is a gram-positive rod arranged in chains. That's a very important point, right? It is non-motile, whereas other members of this particular genus are motile. And it is aerobic as well as it is spore-forming. This is also a very important point, that it is spore-forming. Now, it is the only bacteria with a polypeptide capsule. It is the only bacteria with a polypeptide capsule. Capsule is not the unique point. Its polypeptide nature is actually the unique point, because every other bacteria is having a polysaccharide capsule. But in the case of Bacillus anthracis, there is a polypeptide capsule. And that particular polypeptide capsule is made up of the amino acid D-glutamate, poly-D-glutamate, right? And we know that the capsule is antiphagocytic. Second, we would be discussing about its habitat. It is found in the soil and infects the cattle, sheep, horses, human farm workers at risk. This would be our further discussion. We would not be discussing it here. First of all, now we have discussed about the disease. It causes anthrax of three types: pulmonary, cutaneous, GIT anthrax. Features, we have discussed. The main two important features are that it is a gram-positive rod arranged in chains and it is a spore-forming bacteria. Second and third, now we would be discussing about its transmission, that how is it transmission is in three different mechanisms. The cutaneous is skin related, and the pulmonary is lungs related, and the GIT is stomach, intestine, and all the other different parts of the GIT. If we talk about the cutaneous anthrax, the spores in the soil enter the wound. For example, if a person is having a wound and it is again contaminated with any sort of soil which is having spores of Bacillus anthracis, that particular person would be having cutaneous anthrax. Second is the pulmonary anthrax. The route of transmission is the inhalation. That particular person inhales the spores via the animal hair or wool. And this is most common in people who are farm workers or are having cattle, sheep, goats, and some people who are working in the slaughterhouses or working in the tanneries. Second and the third one, now the third one is the GIT anthrax. The GIT is food related, so spores are ingested via the contaminated undercooked meat, right? So the question arises that how is it causing the disease? How is the mechanism of action working? So this is the pathogenesis. Bacillus anthracis actually produces a toxin which is known as anthrax toxin. Anthrax toxin is having two components, main components. In some areas it is written it is having three components, and some areas it is written that it is having two components. The two components are the edema toxin and the lethal toxin. Edema toxin, the name itself suggests that it would be causing edema, and the lethal toxin is responsible for the tissue destruction, inhibition of the cell growth. This is just an additional point that the anthrax toxin is encoded on one plasmid and the polyglutamate capsule is encoded on the different plasmid. First of all, we must be knowing that, as I mentioned earlier, that this particular anthrax toxin is having three components. Here there are two written. The anthrax toxin is having three different components. One is the edema toxin, the lethal toxin, and the protective antigen. If we talk about the basics of every exotoxin in this particular disease, as we have mentioned that there is an edema toxin and a lethal toxin, each toxin is having two different components or subunits. One is the A, one is the B. Collectively we say AB configuration. The A is the active subunit or the catalytic subunit, which is having the enzymatic catalytic activity. And the second one is the binding subunit, which is also known as protective antigen because it causes the binding of the toxin to the cell. Right? If we talk about now the edema toxin, as the name is itself suggests that it would be causing edema. So edema is actually accumulation of fluid. As everyone knows that it is the accumulation of fluid. But how is the fluid being accumulated? This toxin causes the activation of adenylyl cyclase, as a result of which the CAMP increases and that particular pathway takes place, and there is excessive fluid secretion from the cell to the extracellular environment, as a result of which edema would be occurring. So this is the behind the scenes of the edema. Moving on to the lethal toxin, as mentioned earlier, that it causes cell growth inhibition and the cell destruction. So lethal factor is actually, or lethal toxin is actually a protease. Its mechanism of action is that the protease inhibits or cleaves the phosphokinase, as a result of which the cell signaling pathway is inhibited. The cell signaling pathway that is inhibited is the MAP kinase pathway. MAP actually means mitogen activated protein kinase pathway, as a result of which there is inhibition of cell growth. So this was the reason. Now I have not discussed about these two lines. Now it would be better if I would tell that this binding subunit binds the toxin. So what is its mechanism of action? It binds to the cell and it creates pores in the cell membrane, and it allows these two factors, the edema toxin and the lethal toxin, to enter the cell. Now you would be thinking that why is the name protective antigen, and it can be asked in viva that what is the reason that it is called a protective antigen, even though there is nothing as protective, because it is binding to the cell and it is creating pores and it is allowing the toxins to enter the cell and it is causing the disease. The name protective is due to the fact that if antibodies are made against this particular antigen, then we can protect against from the disease. Actually, it means that we are having a protective antigen, right? Its mechanism of action is here. Done. If we create antibodies against it, we can protect the person from the disease. So that is why it is called protective antigen. So now we have discussed the pathogenesis, and this is an MCQ point that edema toxin, since it increases the fluid secretion, its action is similar to cholera toxin, right? Moving on to the clinical findings, which are actually what we want, because in any case of scenario, we are given with the clinical findings and then we have to do the diagnosis. As I mentioned earlier, there were three types of anthrax: the cutaneous anthrax and the GIT anthrax and the pulmonary anthrax. Cutaneous is skin related. The main thing that we will be seeing, the main clinical finding, is a black eschar. This black eschar is actually, this is a printed diagram, so I would be making like this. For example, this is a central area of necrosis surrounded by this light area or reddish area of edema, right? So it is actually, as I mentioned earlier, that spores enter the skin through the cuts and abrasion. The spores vegetate and the bacteria grows. Now the question arises that why is the spore germinating? Actually, when there is a cut or when there is an abrasion in the skin, the blood vessels there are ruptured, and this particular wound or abrasion is a avascular area. Avascular area, right? And there is actually some sort of necrosis. And avascular area and necrosis promote the germination of a spore, as a result of which the toxins are produced and all that this mechanism of action occurs, as a result of which we get painless black ulcers. Painless black ulcers, this is due to lethal factor. And edema that surrounds the black ulcer, it is due to the edema factor, right? Now, if we talk about the GIT anthrax, as I mentioned earlier, it was due to the ingestion of the contaminated undercooked meat, and the spores are ingested. And whenever GIT is related, you must be knowing vomiting, abdominal pain, and here the bloody diarrhea is also occurring, and in severe cases septicemia is also occurring. This is not that much important, but you must be knowing about the reason and the mechanism of action, right? Moving on to the pulmonary anthrax, which is very, very much important, and it is also having another name which is known as Wool-Sorter's disease, which is an MCQ point, right? Pulmonary, as I mentioned earlier, that it is via the inhalational route. So one might think that it is via the inhalational route. So if a person is having spores in the lungs, and if that particular person will cough, and the person sitting in front of it will also get contaminated, and this would be communicable? So the answer is no. Inhalational anthrax is not communicable. So this is a reason that you should be knowing that why is it not communicable, because when the spores are inhaled, they no doubt go into the lungs, but they do not remain in the lungs. If they were to remain in the lungs, it might be communicable, but they are in the lungs, they immediately go to the mediastinal lymph nodes, as a result of which mediastinitis occurs and mediastinal widening is seen. This is an important viva question, that why inhalational anthrax is not communicable. It is due to the fact that spores, when inhaled in the lungs, they immediately go to the mediastinal lymph nodes and cause mediastinitis, and they do not reside in the lungs, right? This is the reason. Now, the symptoms and the clinical findings that we see, the one is the mediastinitis, that is actually seen as mediastinal widening, that is a telltale sign. And there are also flu-like symptoms, substernal pressure and dry cough. As we all know, since the spores went into the lungs are also infected, and the dry cough is occurring, the substernal pressure, flu-like symptoms, and the pulmonary hemorrhage also occurs, bloody pleural effusion, the mediastinitis is occurring, and the shock and the death. Now, we do the x-ray of that particular person. Normal x-ray is this. As you all can see that this is the heart and everything is normal, right? Now here, the widened area that you're seeing, this is the mediastinal widening. And mediastinal widening is an important diagnostic criteria of pulmonary anthrax. Now, if we remember something, that mediastinal widening is mainly due to aortic dissection. For example, if a surgeon or if a physician sees this type of x-ray, he would say that this particular person is having aortic dissection. But aortic dissection would not be the reason if a person is also having flu-like symptoms, dry cough. If a person is having flu-like symptoms, dry cough and mediastinal widening, then the pulmonary anthrax is the diagnosis. Mainly due to aortic dissection, but if flu-like symptoms is also present, then pulmonary anthrax. This is the reason of the clinical findings of the pulmonary anthrax. Moving on, this is also a point that has been written, that hemorrhagic mediastinitis, as we all know that when into the lungs, they go into the mediastinal lymph nodes. So hemorrhagic mediastinitis, and they also can go to the meninges. The meningitis are life-threatening complications if not treated, right? Moving on to the lab diagnosis. So first of all, we would be taking the specimen. For example, if it is a pulmonary anthrax, we would be taking sputum, and if it is any sort of GIT anthrax or cutaneous, if it is cutaneous anthrax, then we would also be taking fluid from the vesical fluid from the cutaneous lesion, and the blood and CSF specimen are also taken. When we culture it on blood agar, we see gray-white colonies. That is a lab diagnosis of the Bacillus anthracis, the gray-white colonies with wavy edges. They are non-homolytic colonies. The gram staining, as we all know, that they are the gram-positive rods. And one more point that everyone should know, that they are arranged in the chains, right? That's an important point also for OSPE. On microscopy, we see gram-positive rods arranged in chain. It was written here. And we said that it was having a polypeptide capsule, and if we want to see that capsule, we use a particular stain which is known as McFadyen stain. Google the pronunciation. I think I'm not pronouncing it correct. And for example, if we want rapid diagnosis in case of a bioterror attack, before going further, we must be knowing that what is a bioterror attack. Bioterror attack is actually when the spores are sent to any particular country in sort of attacking that particular person. For example, if I'm getting a letter and I'm opening that particular letter, and in that letter the spores were already present, and I inhaled those spores and I just got the disease. So in those particular cases, in case of bioterror attack, the rapid diagnosis is required. And for rapid diagnosis, we have two tests. One is the PCR, polymerase chain reaction, and one is the direct fluorescent antibody test, which detects the antigen of the organism in the lesion. The serologic test is ELISA. That's a little bit of cramming, and that's just a note that to detect the antigen, serology, immunofluorescent staining is used. Treatment. We are having a drug of choice, which is ciprofloxacin, right? If it is not present, the alternative drug would be doxycycline. And there are no resistant strain. as we have studied that a lot of bacteria are having resistance. So there are no resistant strains have been isolated up till now. For prevention and for prophylaxis, we require ciprofloxacin and doxycycline, and we also give vaccination and immunization. That's also just a point that you should be knowing. If you want details, then you should be remembering that within a period of 1.5 years, that is 18 months, you should be giving 6 doses of vaccine to the person. An annual booster should also must be given. Now this particular point should be remembered because this is something that is unique and important to know. If you want to prevent the disease, and if you know that an animal died of anthrax, you should not bury that particular animal, because when you will bury that particular animal, the spores will be present inside the animal and the soil will also be contaminated. You should not bury such animals, otherwise the soil will become contaminated with the spores. The alternative and the correct way to prevent the disease is that you should incinerate those animals. This is burn those animals. Burn those animals so that there would be no animal, no spore, no remains, no contamination, and prevention only. So this is it about the Bacillus anthracis. After a lot of information that has been given, it would be better if it ends with some visualization. This is a picture of Sketchy Micro which depicts everything, which tells actually everything about the Bacillus anthracis as well as the Bacillus cereus. Bacillus cereus will be discussed in the next bacteria. Here we will be discussing about the Bacillus anthracis. As you all can see that this picture is of some sort of Vikings, and this is the ship, this is the camps, and the boats are sailing, and all that scenario has been created, right? If we begin with the Bacillus anthracis, as you all can see these are the ships. These are rod-like ships sailing. They are arranged in the form of chains. So as you all can see, these are the gram-positive rods arranged in chains. So Bacillus anthracis is gram-positive rod arranged in chains, right? So these are the Vikings that you can see, right? They are having a capsule on their head, cap on their head. So this capsule tells that it is encapsulated, and since they are having a leather jacket on them, which is indicating some sort of protein. So this leather jacket is indicative of protein, means that the capsule is made of proteins, and this D belt tells that this is poly-D-glutamate. Clarified? Now we know that the bacilli produce two toxins. One was the edema toxin or the edema factor, and the lethal toxin or the lethal factor. As you all can see that this is a shield. If I zoom it, this is the shield on which EF is written, and this is a shield on which LF is written. This EF is the edema factor. So what it causes, as we all know, that it causes increased CAMP pathway. So this increased CAMP pathway, these camps, as you see, CAMP, camp, pathway, this is a pathway of camp. So increased CAMP pathway is being shown here. This is the edema factor, edema factor's mechanism of action, right? If we now talk about the lethal factor, it caused the inhibition of MAP kinase. So this is a Viking that is burning a MAP, inhibiting a MAP. So it inhibits the MAP kinase pathway. So this is the representation of inhibition of the MAP kinase pathway. One thing I forgot to mention was that, as you can see that here are the walnuts are present. These walnuts are indicating that Bacillus anthracis is spore forming, right? And this is an air blower. It means that it is preventing the fire, right? So it is an obligate aerobe. Now as you can see that this axe is lung shaped, so it affects the lungs, and this blood indicates that it causes hemorrhagic mediastinitis, right? And now as you can see that this particular ring in which they are standing with a black base, so this is a black eschar. So as I mentioned earlier that the black eschar has a central tissue necrosis with an edematous ring surrounding. So this is a ring of edema, this is the central tissue. So this indicates that Bacillus anthracis in its cutaneous form causes black eschar, right? If we now talk about the pulmonary, so this was the lungs, as I mentioned earlier. These are the lungs. It causes pulmonary hemorrhage, and this is a sheep, so it is caused by the sheep wool sorter disease. And if you can see that this ship, for example, this is an X-ray, and this widening that you see in the middle, this is indicating mediastinal widening, right? This is the mediastinal widening. And one more thing about the treatment, as you can see that this is the flower, this is a bicycle wheel. So flower for fluoroquinolone, in which ciprofloxacin was the drug of choice. And what was the alternative drug? Cycle, the doxycycline, right? So this is an indication of doxycycline, the fluoroquinolone. This is encapsulated protein in nature, poly-D-glutamate, edema factor, causes the increase in the CAMP pathway, lethal factor, inhibits the MAP kinase pathway. This is the black eschar edematous ring, central tissue necrosis. This is the sheep causing the pulmonary anthrax, wool sorter disease, and we see on X-ray that there is the mediastinal widening and the lungs are affected, the hemorrhagic mediastinitis is occurring. These are the gram-positive rod features, that they are the gram-positive rods arranged in chains, and they are spore forming, as you can see here, and they are obligate aerobes, right? We have discussed everything. And the one thing that has yet not been discussed is this particular Viking, which would be discussed in the next bacteria. And this brings to the end of Bacillus anthracis.